Hello guys, welcome back to the video of Medical and YouTube channel. So this is me Akash Jet. So we are going to study Wilson's disease today. Okay, Wilson's disease is read in biochemistry also, and it is also read in liver pathology. Means it is in pathology second year also. So it is a very important topic. Please stay alert. Okay, Wilson's disease. It is an autosomal recessive disorder caused by mutation of ATP7B gene, resulting in impaired copper excretion in the bile and failure to incorporate copper in the celluloplasmin. Okay, let me explain in a simple way. See, you can see there is copper here and there is alpha-2 globulin. Both combine and form the celluloplasmin. Celluloplasmin is one of the plasma protein. So for combination of copper and the alpha-2 globulin, we require ATPase enzyme. ATPase enzyme. So this ATPase enzyme is coded by ATP7B gene. Okay, when there is mutation in the ATP7B gene, there is absence of this ATPase enzyme. So thus copper and alpha globulin cannot combine and there is no formation of celluloplasmin. Thus more and more copper is increased in the serum and it get deposited in the different parts of the body. Okay, understood? The enzyme produced by ATP7B gene, that is ATPase. So you can read it now. Wilson's disease, mutation in ATP7B gene present on chromosome number 13. ATP7B gene codes ATPase enzyme. Okay, encodes copper transporting ATPase responsible for incorporation into alpha-2 globulin to produce ceruloplasmin in the liver. Okay, so copper accumulation in eye, liver, and primary there lies in the ceruloplasmin level in the circulation. There is rise in ceruloplasmin level in the circulation. The accumulated in the causes toxic liver injury and spillage of non-ceruloplasmin bound copper from the liver into the blood deposited into extrahepatic tissues such as brain and eyes. Okay, so as ATP7B gene is mutated, there is no ATPase enzyme and thus there is decrease in the ceruloplasmin level and increase in the copper content, and that copper is deposited into liver, brain and eye. Okay, that it is written. Okay, in young patient symptoms of liver disease is seen, and in older patient neuropsychiatric symptoms are seen. The symptoms of liver disease because there is deposition of copper in the liver, and neuropsychiatric symptom because of deposition of the copper in the brain. And one of the most important characteristic feature of this Wilson's disease is formation of Kayser-Fleischer ring. Okay, Kayser-Fleischer ring in the deposition of copper in the cornea. Deposition of copper in the cornea is Kayser-Fleischer ring. Remember, I will add a picture of Kayser-Fleischer ring at the end of this video. Please watch it. Okay, and the neurological symptoms like tremors, emotional imbalance, difficulty with handwriting, and Parkinsonism. And the symptoms of liver failure like hepatomegaly, ascites, and cirrhosis. Clinical features of this Wilson's disease. First is hepatic, means the copper is deposited in the liver, so there is formation of cirrhosis. Okay, for cirrhosis to happen, there is a necessary for fatty change and also acute and chronic hepatitis. Okay, you know process of cirrhosis. First there is hepatitis, then there is steatosis, then at last it is cirrhosis. Okay, 3 stages. Next there is Mallory Dank bodies are seen in cirrhosis. Okay, Mallory Dank bodies. Next you can next coming to the CNS, basal ganglia is involved primarily. There is tremor and chorea as the copper is deposited in the brain that is in basal ganglia. There is tremor and chorea are the symptoms seen. Motor deficit and sensory involvement are the characteristically absent. Motor deficit and sensory involvement are characteristically absent. Okay, there is only tremor and chorea and there is no sensory or motor deficit. A person can move his hands and he can feel sensations, everything. Next, ocular involvement, that is formation of a Kayser-Fleischer ring in the cornea, and on the lens there is sunflower cataract. Okay, now coming to the diagnosis of this Wilson's disease. First we can do the liver biopsy and quantitative copper assay. On quantitative copper assay, you can see there is increase in the amount of copper content. Okay, which is a gold standard for diagnosis of liver biopsy with quantitative copper assay. Concentration of copper in the liver biopsy sample that is greater than 200 milligram per dry weight. Okay, as copper concentration is increased in the liver, that is greater than 200 milligram per gram of dry weight of the liver. Next, we can do serum ceruloplasmin level, which will be low, as there is no ATPase enzyme. Copper cannot combine with alpha globulin, and there is no ceruloplasmin, decreased ceruloplasmin formation. Therefore, we can see low ceruloplasmin count. Next, urine excretion of copper in urine is increased, and formation of Kessler-Fletcher-Ling in the cornea, and we can do DNA haplotype analysis. Okay. This is the Wilson's disease, and it is the most important question for theory exams like board exams. Okay. Please like, share and subscribe. See you in the next video. Until then, Jai Hind, Jai Bharat.